is basically the exogenous participation of stem cells in glomerular repair.
Good morning, and thank you for inviting me to this presentation. The primary part of my presentation is going to be to explore our work with exogenous mesenchymal stem cells, and then I'll talk some about what the induced pluripotent stem cells could do for us. We know, this study of the stem cells is a growing field, but in the kidney, especially in the glomerulus, it is lag behind, and this is where we come in. Our laboratory has been engaged in the study of mesangial repair and injury to the mesangium produced by the two diseases you've heard before, AL amyloidosis and light chain deposition disease. We use exogenous mesenchymal stem cells to repopulate the damaged mesangium and observe their differentiation into mature mesangial cells, which Dr. Teng attempted to show. We reproduce the mesangial injury, or we produce mesangial injury by exposing the glomeruli to glomerulopathic monoclonal immunoglobulin light chains obtained from patients with either AL amyloidosis or light chain deposition disease. These abnormal light chains can produce damage either by, in the case of AL amyloid, lysis of the mesangial matrix with replacement of the damaged matrix by amyloid, or increased matrix by deposition of abnormal light chains from light chain deposition disease, depending on their physicochemical characteristics, because they are conformationally different. Our unique experimental models for mesangial injury allow us to explore the use of exogenous stem cells for mesangial repair of the glomerulus, and this is of the damages produced by these abnormal light chains that I mentioned. We know from work of others, and Sugimoto in particular, that he demonstrated that when you transplant wild-type bone marrow into irradiated mice that there's recruitment of bone marrow cells which differentiate as podocytes and mesangial cells, leading to partial restoration of type 4 collagen, and particularly the alpha-3 chain, and emergence of the alpha-4 and alpha-5. So we know that there's a reparative condition that can happen. And this has been mentioned before, but where do these mesenchymal stem cells come? Well, there's the exogenous I'm going to show you about, and then, of course, the bone marrow. And then there's the niches in the kidney, which Dr. Romagnani talked about. I mean, there's in the tubules, they're in the Bowman's capsule, etc. And these are the stem cells growing in a plate. And they're very primitive, as it was mentioned before. These are mostly primarily nucleus, very little cytoplasm, only a little rough endoplasmic reticulum, and some ribosomes. So we have two models to study these diseases. We have the in vivo and the in vitro models that Dr. Teng explained. And these are used to observe what happens when we first cause the damage into the cells and then try to repair it. The sequence of events that lead to mesenchymal stem cells identifying the site of glomerular damage, eliminating the remnants of damaged mesangium such as apoptotic cells and extraneous matrix, either in the light chain deposition disease or AL amyloid, and finally differentiating into mature mesangial cells, which then lay down new mesangial matrix, and we have observed this. Part of our in vitro studies includes our observations utilizing this equipment that Dr. Ten showed. It's a multi-dimensional system that shows us the cells growing and moving. And we can keep this going for as long as 14 days. Information obtained by each model system has allowed us to better understand the exogenous mesenchymal stem cells and how they participate in the repair of glomerular damage. This is the ex vivo system that, as you know, we can use it for rat kidneys but for mouse kidneys, and of course the renal arteries cannulated, and then we keep pressures and everything to know that everything is physiologic. And then, of course, the ureter is also cannulated to obtain urine for studies. In the ex vivo platform, this is in AL amyloid, and we can see the disease has been established here with amyloid being formed in the mesangium, and we can see here, this is Congo red, which is sustained for 100 years at least as being in use, and this one, when we polarize it, is apple green birefringent, so we know it's amyloid. And here we have now infused the mesenchymal stem cells, which then go to the area of injury. And this is an enlarged in the ex vivo model. First, we put the abnormal light chains into the kidney, allow for the disease to happen, and then the mesenchymal stem cells come in, and you can see them that they are in the glomerulus in here next to the amyloid trying to do their work. The mesenchymal stem cells, we label them with a red dye, and after creating the injury, this is in the light chain deposition disease, create the injury, we have the nodules, this is now the mesenchymal stem cells have been labeled with red, and you can see them here, and then in time, how they spread into the mesangial areas where the lesion is. We have observed this the cells also by electron microscopy to see how they interact with the mesangium, and this is what I'm going to show you. The disadvantage we have with the ex vivo model, which is the kidney that's being maintained physiologically for 96 hours, is that it's only short time. And also we can see the snapshots of the process, but this is how it looks by electron microscopy. This is now the glomerulus, and these are the mesenchymal stem cells. You see how much primitive and simple they are compared to the mesangial cell. And as you see how they enlarge, you see the large nucleus, the little bitty cytoplasm. And as they get and start working, they do have more cytoplasm. Here they come. This is the same mesenchymal stem cell. It's now hugging, like with is putting this pseudopodia around the debris to try to destroy it. And here we see them with now have these macrophage characteristics almost. These are the mesenchymal stem cells, and they have these lysosomes, and they have part of the debris that they have been acquiring in this. This is the 60 image that Dr. Teng was mentioning, and this is a very fancy equipment that see the cells in different aspects. You can have an X, Y, and Z, and even a lambda, and then different colors can be applied. These cells, the advantage of this system, we can keep the cells going for 14 days, and they're taking pictures continuously. However Dr. Teng sets it up, a few minutes, 4 minutes, 10 minutes, whatever you set it up. And that's how we have seen our cells growing and differentiating. These are cells here, and I think this one's been presented before, but these are the human mesangial cells that are growing on matrigel. And these are the little nodules that this is the light chain, this is amyloid forming. And this is these waxy nodule that has formed with this is just formed by amyloid. And when we put the mesenchymal stem cells tagged with green, they come and flock to that area of the nodule and try to destroy it. And this picture has been shown by Dr. Herrera before, and you can see this is the amyloid and the mesenchymal stem cells coming and surrounding the area. By this lysotracker red, these are the mesenchymal stem cells, and this is the phagocytosed material. These are remnants of the mesenchymal stem cells and the amyloid, and they're just destroying it. And now I have a sequence here, so you can see how this is that same picture, and as they work in time frame, they destroy that amyloid, internalize it, and then look at these pictures. This is the mesenchymal stem cells, and now they're kind of becoming different, and they're extending their cytoplasm is becoming more abundant, and they're replacing. These are the mesangial cells now that they have differentiated into. We have that in video as well. In the light chain deposition disease, which is the other disease, this is the same process. This is the cell culture model. First, we induce the disease, and these are the nodules, and then the mesenchymal stem cells are put in, and they're labeled with red here. And you can see in a sequence, I'm going to show you how, they have gone from out here, in here, into the nodules. See how irregular this is, because this light chain deposition disease produces nodules, like in diabetes, if you happen to know. And this is the mesenchymal stem cells in red coming to the area. Now, what happens with light chain deposition disease is that the mesenchymal stem cells by themselves cannot really take care of the nodules because there's tenacin, as you've been mentioned before, and this is protein that has the shape of an octopus that is very tenacious and is difficult to destroy. So curcumin enhances mesangial repair by the stem cells, making them work better and also priming the stroma for destruction. And these are red mesangial cells with no curcumin. And you can see this is the material they have to attack, and these are the mesangial stem cells, the mesenchymal stem cells. In this sequence of events, you see this is the nodules, and you were shown this before, but what happens is that the mesenchymal stem cells by themselves cannot touch the nodules. But when you add curcumin to the system, which first is we induce the lesion by putting the abnormal light chains, then we put the curcumin with the cells. And now you will see in the sequence of events that the nodules are destroyed or never formed, actually. And here we have the mesenchymal stem cells and the nodules. So the only way to destroy these nodules or try to repair a lesion like light chain deposition disease or diabetes is by, we have found, with the help of something like curcumin. Curcumin is in the literature known that it does reduce extracellular matrix in keloids. For example, you're going to treat a keloids, curcumin does, keloids have tenacin in them, very hard to treat. But with curcumin, these can be ameliorated. Curcumin also induces apoptosis, and this has been published. And in our work, we've seen it enhances migration of the stem cells to the area and then helping for destruction. And to study this, how it is thought that it cleaves the connection between integrins and the extracellular matrix and sub. But we have seen, and we're going to study further, that it primes the extracellular matrix in an unknown fashion and helps the destruction of these very tenacious nodules. In the ex vivo, remember the ex vivo model is the one we have the kidney being perfused, keeping physiologically. This is without curcumin, and you can see that the cells, this is in the light chain deposition disease case, and the cells come in and very little. But when you prime it with curcumin, then you get the mesenchymal stem cells attacking those nodules. They're tagged in red. In conclusion for this part is that, our research efforts have led us to a much better understanding of the pathologic mechanisms involved in AL amyloidosis and at light chain deposition disease, creating a model of mesangial damage that we call the altered mesangium that we can there try to repair. In vitro and in vivo testing are in progress to further develop our model for therapeutic strategies to target key steps in the pathogenesis of these disorders. So, moving the therapy from a molecular level to the patient, which is what we ultimately want. And the stem cells provide a novel avenue to accomplish mesangial repair, but much work still remains to be done to take it to the clinical practice. Where are we going from here? And we have been exploring the possibility of using induced pluripotential stem cells in our animal models. And as you know, the pluripotential stem cells are a type of stem cells that, like the induced ones, cells that can be generated from terminally differentiated or mature cells. And this is a very exciting field. Let me see if I skip one. Yeah. As you know, Dr. Yamanaka from Kyoto is the pioneer of this revolutionary technology it allows experimentation and ultimate repair and treatment of conditions. Advantages of iPSCs, besides, number one, not having to destroy or manipulate an embryo, which is something that is, for us, is very important. And number two, that these stem cells could be made patient-specific and therefore used for treatment, like it's been mentioned this morning. Dr. Yamanaka and his group in 2006 introduced this method of inducing pluripotency using a retrovirus vector. And by this, he delivered 24 genes that would stimulate the embryonic stem cell specific gene X15. These cells will then be isolated by antibody selection. What happens upon the delivery of these genes, the cells that came out resembled and tested like embryonic stem cells. And for this, he got the Nobel Prize. But what happens is that his lab proceeded working to minimize these 24 factors into 4 and make it simpler. And now, we know that from OCT4, SOX2, CMYC, and KLF4, there's reactivation of FBX15, so the cells become pluripotential. So in this group, no, I'm going to skip that for interest of time, but, other things. For the first time in 2007, iPSCs were created from human adult cells from 2 groups, a group from Dr. Thompson's lab in the University of Wisconsin, which was published in Science, and a group from Dr. Yamanaka's group that was published in Cell, and that human cells could be now made in pluripotential. The problem with generation of iPSCs is that it's slow and inefficient process, very, very inefficient. And there's many groups in the world that are working to make this process efficient so it can be used. So, and by this system, it's very inefficient. You see 0.01 to 0.1 efficiency, so it's very, very difficult. But upon introduction of the reprogramming factors, cell colonies that resemble pluripotential stem cells can be produced. So because iPSC delivered by vectors, viral vectors, can be oncogenic, then there's all this work being done to reduce this. And in 2008, a technique came out where they could remove the oncogenes after delivering the transforming factors, removing the oncogenic. And a lot of work has been done. In 2009, it's been published, the iPSCs can be generated without alteration of the genome by using proteins that could deliver the same transforming factors. And this is, there's a lot of literature about it, but basically there are cells, certain proteins like polyarginine anchors that could deliver cells or the factors into the cell. So there's many ways that people have been exploring how to do this without damaging the genome. And viruses have been used that do not integrate. And transfection of modified messenger RNA that can encode for the reprogramming factors. So it's an amazing kind of body of work. And there's molecules, small and large, so chemical things can also be induced. Proteins and also chemical induction for iPSCs. So the important thing about this is that these compounds can be synthetic or natural. There's an amazing interest in this thing. So what I wanted to, for the interest of time, much work has been done, but a lot needs to be done for it to be made available for patients, which is ultimately what we want. We want to make it safe, efficient, and useful for the treatment of patients, which is our ultimate goal. And thank you very much.
So we have about 15 minutes for questions, so if we can bring all the speakers to the table, and then we can open this session to questions. Please, if the speakers could come to the table. So any questions from the audience for any of the speakers? Yes, sir.
I have questions for Dr. Romagnani.
Do we have a microphone that we can circulate around?
Yeah.
There we go. Great.
First, I just wanted to say I really enjoyed your talk. It's very elegant work. My questions are, given that you now are identifying these progenitor cells in the kidney, are you pursuing any efforts to expand them, to make more of them? And a related question is that you're using sort of the promoters as reporters for progenitor cells. Have you done any I know there's been labeling work done in the past. I'm just wondering if you've done any to ask how does things like BRDU labeling layer on top of your biomarkers?
Okay, so what you have heard today is basically the last part of this work that is the result of the most recent 2 years. But then we have published many papers in previous years where we have mostly characterized these cells in the human kidney tissue. So, and we started all this work just purifying the population from human kidney tissue using CD123 as a marker, then functionally studying it. I mean, we can amplify it, this population, we can clone it, and then we establish techniques to differentiate the clonal progeny of these cells into different cell types of the kidney. We injected them into skid mice with different type of disorder, glomerular disorder, tubular disorders, and we saw that they engraft and become different type of kidney cells. So, I mean, this was published in 7, 8 papers in JASN and in Journal of Experimental Medicine. And then, so the most recent part was we need to find exactly the same population in the mouse kidney, and these are the data that you have seen and for which we have established the mouse model. But all the part on the culture, on the characteristic of these cells in culture, and particularly in human, is largely known and very well established.
Any other questions?
Maybe jumping off the last speaker,
Excuse me.
So you showed in that confetti mouse model that you had in early time points that all the cells of the glomerulus were labeled. I wonder if you thought there was a transit amplifying cell that was propagated from an initial cell, if there's a long-term population of stem cells there. And then additionally, in the zebrafish, to go off your question of expanding that renal progenitor population, there's an HDAC inhibitor that can expand the PAX2A population in zebrafish.
This is a very good question. I mean, the paper, we have not checked the population in the zebrafish. The data in the zebrafish are published by Alan Davidson. He published the paper in Nature 3 years ago, yes, where he described this population of cells that can be transplanted and generate the whole nephron in the zebrafish.
Yeah, LHX1 positive population, I think, yeah.
Yes, exactly. And, well, the population that is in human is more made of an heterogeneous population of progenitors with different commitment, okay? Still, these cells are all characterized in human by one single marker that can clearly distinguish the cell from all the other, that is CD123. So you can recover them, and then you can get different clonal populations. Some of them still keep the capacity to differentiate in epithelial cell of the glom or in epithelial cell of the nephron, because these are epithelial progenitors, okay? They do not generate all the interstitial and mesenchymal part of the kidney. And they have a distribution in the human kidney that is extremely similar to the one that has been described not only in the zebrafish, but also, for example, in the insect. So they are localized at the point of connection among the glom and the tubule, and also scattered within the nephron. And, but it's different from the story of the fish, because there it's a totipotent population somehow that has been described. But you know that the fish still keep the potential to generate new nephrons, something that mammals cannot do anymore. And the reason why it's not known, probably it's because the fish has a few nephrons, and mammals have many, so there is no need anymore of something like this, but more to replace small portion of the nephrons that may be injured.
So to jump off a little bit more there, so you brought the glomerulus and the tubule, that position where there's glomerulus and the tubule, that population of cells reside. Do you think that's a hypoxic environment where those cells may reside there? Maybe a niche?
Yes, potential? I think so, because most of the tubular cells of the tubular compartment, I mean, they are distributed in specific areas of the tubule, but most of them are localized in the S3 segment, which is the most hypoxic area of the kidney, and this is the segment where it's really needed, because it's where you get the injury during acute kidney injury.
Thank you very much. It was beautiful.
I have a question regarding homing. I think it's of general interest. Is this homing mechanism related to a tissue-specific signaling, or are those general signals that relates to inflammation? That's my question.
I actually think that in the case of our model, the damaged tissue and the milieu around that damaged tissue is an important signal for the mesenchymal stem cells to go there. Whether that is a very specific signaling at the level of the mesenchymal, I don't think so. I think that this signaling could be more generic.
If it's generic, then I can imagine that other inflammation in the body could actually distract the whole procedure. I mean, the mesenchymal cells may be confused and they may go somewhere else. So I don't know if somebody thought about that.
That's a very important point, and obviously what you're saying is interesting in that if you give too much confusion to the mesenchymal cells, they don't know where to go. One of the most amazing things in this research is the fact that they go where the damage is. It's amazing.
Yeah.
That was something that we saw and we couldn't believe it, that it was so specific where these cells ended up. You know, I would think that, yes, maybe there's something right there in that damaged mesenchymal that says, come over here, don't go somewhere else. What it is, I don't know.
Maybe another question, but tenascin, I mean, is there something specific in mesenchymal stem cells that could degrade tenascin, this specific resistant protein?
Actually, the problem is that the mesenchymal stem cells have a problem with tenascin. They just cannot dissolve it, cannot break it down.
We have
And remember that tenascin not only accumulates in light chain deposition disease, but also in diabetic nephropathy. It's the protein that is in those nodules. So mesenchymal stem cells need help. They just cannot go there and take care of the tenascin. That's where the curcumin comes into place. There could be other things that could help them. We thought about metalloproteinases. I mean, we know tenascin is degraded by MMP7. So one of the things that we may want to do with the microbe manipulator is provide to those nodules MMP7, help the mesenchymal stem cells come in and degrade and repair. But they have trouble. By themselves, they cannot do it.
This is for Dr. Herrera. I'm curious, I mean, I'm actually struck by this. Maybe this is the first time the two groups have sort of been at the same podium together. But have you considered using the CD133 positive cells in your model?
And we have as a way to trace stem cells going into the glomerulus. In our light microscopy pictures, we stain the cells for CD133. So we have. We also use it.
So I'm asking a different
Yeah,
question.
yeah.
Not CD133 out of the bone
Okay.
marrow, because there are lots of CD133 cells in the body in many different
Right.
parts. I mean these cells that are being identified in the kidney, in the location where the damage is occurring.
Yeah, we have looked for them. They're there. You know, when you have tubular damage, for example, CD133 cells immediately come out. So the tubules work with the stem cells much better than the glomerulus does. But can you get 133 cells in the glomerulus where the repair is happening? Sure you can.
She's talking about the native ones in the niches.
Oh, in the niches, you mean? You mean the
Yeah.
niches cells?
The cells in the niches. Will they come in? I think they're overwhelmed by the disease.
Yeah, I think once we create the damage of the degree that we create, the normal physiological ways of trying to fix it are impaired. It's just too much for what's already there, and that's why you need something from the outside.
Any other questions?
Well, if there are no other questions, I want to again thank Dr. Parolini for donating her time. She will have the keynote address tomorrow. She will start at 8:30, so please be here because she's going to incorporate the information in that keynote address.
She was kind enough
And I
to
thank
offer.
all of the attendees, and hopefully you learned something from this workshop. Thank you very much.